Welcome, viewers, to this new episode of BeyondPhrenology. We are extremely fortunate to have with us Professor Gerald Loeb from University of Southern California. Uh, it's hard to, give a brief introduction for what he has done and what he's known for. He is an MD, but he is a great inventor. He has more than 75 patents with respect to, pacemakers and prosthetic devices, mostly biomedical devices. He's also fellow of the American Academy for Inventors for the same reason. He has hundreds of publications in the field of motor control and physiology, and he has been one of the most fundamental thinkers in the field, especially when we are talking about not just physiology, but engineering approaches to understand physiology, some of the limitations of those approaches, and some of the new ways in which we need to approach physiology if we have to clinically translate our understanding uh, for the betterment of human movement and perception. Welcome, Professor Loeb, to BeyondPhrenology.
Great to be here, Matter.
So, Professor Loeb, I would start. I would like to start with. I'm interested about some of your background. Like you started as an MD, and you started with surgery, and here we are discussing motor control, and most of your work has been, in device development and understanding the body from that perspective. So, a little bit about your early years of education. You have been in the field more than probably like twice my age now. So, where did you find yourself to begin with your career, and um, how did you tread
So,
along this particular trajectory?
yeah, I've chose to go into medicine because I see the human body as essentially a machine that, when it breaks, needs to be repaired. It's a fairly engineering approach to something, and so if you want to fix things that are broken, first you have to understand how they work normally, what they're supposed to be doing, what sorts of interventions are possible, and uh, and then develop technologies that will meet the dysfunctions that clinicians know about. So the first thing in my mind for such a career was to study enough medicine and enough surgical and clinical technique to be able to appreciate the normal and abnormal function of the human body. And alongside that, to find opportunities to get at the basic science of how the thing works, both to learn from what's in the literature and, if necessary, to extend it when we find things that we don't understand, and enough of the technology so that we can identify the opportunities to intervene with technology when things are broken. So
Yeah.
that's pretty much the background. I went to Johns Hopkins both as an undergraduate and in the medical school in a combined program that they no longer have, but it was designed to encourage people to do the kinds of career that I've had, which is to combine science and engineering and clinical skills to do research rather than just follow the existing techniques.
Right. Uh, but what made you specifically focus on spinal cord?
At the time, I mean, most of the things I wind up getting involved in are really accidental opportunities that present
Hmm.
themselves and seem interesting. Uh, the field of neurophysiology was pretty much fixated on spinal cord and sensorimotor control when I started as a student, which is in the 60s. Um, so it was natural to look at the nervous system from that perspective because it's more accessible. You can uh, look at the inputs and the outputs both in a fairly practical experimental way. The actual first research that I did was to develop techniques to interface with neurons at the level of tissue culture.
Hmm.
And it happened that we used as our first cells those that can be obtained from the dorsal root ganglia. Uh, then, because I learned something about microelectronics and neurophysiology, which was a fairly rare combination in the 60s, I got involved in one of the first attempts to build prosthetic implantable devices. And that was actually with the Visual Prosthesis Project, starting at Hopkins and also at University of Utah.
Right. And Utah has also been, one of the, I think, powerhouses of prosthesis research. And interfaces since then, especially with the Utah interface as well when it comes to brain,
Yep.
right? Have you seen? I mean, we have much more advanced prosthesis now, compared to 60 years back, right? Well, so where exactly has been the most innovation or understanding of prosthesis now? Uh, Also, there are different areas of prosthesis, right? We can talk about, for instance, like auditory prosthesis, or even deep brain stimulator is also kind of prosthesis, right? At a neural level, compared to prosthesis, which generally when people think of, they think of an artificial arm or artificial leg. So where has been the most innovation, and what are the some of the things we are still limited by our understanding of
So I think most of the
knowledge?
advances can be related to people systematically applying the biophysical principles of how the nervous system works and how individual neurons respond to electrical stimulation, and how the signals that arise from such stimulation would get processed by the parts of the nervous system that you're trying to modulate. There's been a long history, and I've written reviews about this, of treating electricity as if it's magic
Yeah.
and just sticking wires in and buzzing things in the hopes that you can fix something that isn't working. And occasionally there have been some mild successes that encourage such a strategy. But the successful devices that we see now changing the clinical scene for many of the disorders that you mentioned are the result of systematically applying real science and real engineering, so that devices are designed according to first principles rather than simply lucking in from
Mhm.
empiricism. One of the reasons that empiricism of just trying things and seeing what happens worked is that there were a few opportunities where even very crude interventions resulted, at least occasionally, in very positive outcomes, and that encouraged the field. But in the end, to really do good for a large percentage of your patients, you have to know what's happening and how to intervene systematically.
Okay, so there's a lot to unpack there. Uh, so let's come back to when you say the broken body and to repair, that body. So broken is a metaphor again. When we think of breaking, we think of machines, apart, being broken, which is which we can apply to body under certain conditions. For instance, like say you have a spinal cord injury, or you have sudden breakdown of a neuromuscular circuit. At the same time, for instance, we cannot say that aging is, you know, a breaking of the human physiology. So, so what do you when you said the breaking of the body, what did you point like what were you assuming? And then second, some examples of first principles thinking versus empiricism, where empiricism hits the limit, and you need first principle systemic thinking to have any success.
So by breaking, I'm speaking very generically about all the ways in which a nervous system is no longer functioning in a way that a patient considers desirable. And that may arise from injury or from disease or from genetic abnormalities, um, from reactions to toxins or drugs. Um, and in each case, uh, you have to find what the actual loss is, and then consider whether the function that you need to repair uh, can be addressed with the available interfaces. So you went through a range of very different things that have had very different outcomes. The restoring of hearing and sensory neural deafness using cochlear implants, which I've been involved in for decades, uh, is an example of a situation in which, even though the system is immensely sophisticated at a neurophysiological level, relatively simple interventions produced substantial clinical gain. In fact, the very simplest devices, which were very badly designed, in my opinion,
Mhm.
with essentially no consideration of the underlying biophysics of the auditory nervous system, still managed to produce enough benefit for patients that it motivated the field to develop the much more sophisticated technologies that we have today. Conversely, The visual system, where I've also been involved starting in the 60s, has yet to get a clinically useful uh, prosthesis that's generally accepted and in use because the fundamentals of either the neurophysiology of the retina or the surgical accessibility of the occipital cortex are simply not in favor of producing sufficient uh, capabilities to treat the underlying disorder. So even a little bit of hearing in someone who's profoundly deaf
Yeah.
can transform their lives. But to produce a useful visual image actually requires a much more sophisticated interface. So the threshold of utility is
Hmm.
much higher. Then you take another field like stroke or spinal cord injury, where the disability is locomotion or walking or using hands for manipulation. And there you have a situation in which the loss appears to be just the motor
Right.
output, but in fact, effective use of the motor output requires an immensely sophisticated coordination of
Hmm.
sensory information and motor intention and the functioning of the muscles.
Yeah.
So that's a field where, uh, again, there has been very little in the way of practical consequence for patients with, say, spinal cord injury.
So pacemaker, for instance, would you agree that even pacemaker is an empirical success because it has nowhere close to the kind of complexity which you see in the real pacemaker tissue, but at the same time, it has immense benefit to patients, and that's why we have like now millions of pacemakers functioning, even though they're not close to complexity with the actual pacemaking in the heart.
So there are different levels of function and dysfunction. The first pacemakers were designed and intended entirely for a fatal disorder that consisted of complete atrial ventricular dissociation. That is, that the natural pacemaker was not able to drive the ventricles, and so the heart then results in a very slow, what we call a bradycardia, that's incompatible with life.
Mhm.
And you can restore a more physiological function with the simplest of interventions, a ticker that simply activates the heart at a sufficient rate that blood flow will keep you from dying.
Mhm.
So there's a very low threshold of utility.
Yeah.
You need only a slight function, but the gain of that utility is huge. It's the difference between life and death.
Yeah.
Once you have that, then you can build on it to add more functions. And the pacemakers we have now synchronize with the existing heart rate and demand as your physiological demand changes, they change the heart rate, reproducing a lot of the feedback loop that the natural biological pacemaker could make use of. So there's probably not a lot of room left to improve functionality in pacemakers. For the last 10 years, been doing research on trying to fix some of their shortcomings involved in the longevity of those pacemakers.
Yeah.
As we put them in younger and younger patients, we need them to last for decades instead of years. And for the accessibility, especially in patients who are too small, very young children,
Mhm.
too small to have the typical pacemaker inserted into the ventricle.
Right. So you have a recent patent on a leadless pacemaker, right?
Yes.
So what's the advantage of that pacemaker compared to the previous versions?
So this is another one of those adventitious collaborations that lead one into a field that one never planned to work in.
Hmm.
Uh, about 10, 12 years ago, a couple of clinicians, one a pediatric electrocardiologist, essentially someone who studies heart rates and ECGs, and a fetal interventional surgeon uh, came to my office because they had a great idea for a pacemaker that could deal with a rather rare disorder in that occurs in the developing fetus in utero, in which the pathway between the atrium and the conduction to the ventricle fails to develop or deteriorates for reasons that aren't well understood. It's a relatively rare disorder, but it's essentially the same condition that motivated the first pacemakers for adults: atrioventricular dissociation, bradycardia, congestive heart failure, and death in utero before the infant can be delivered.
Yeah. Okay.
So they had the idea of a pacemaker that could be injected through a 3 millimeter diameter cannula that they used to actually perform interventional surgery in utero on hearts, on fetal hearts, to correct mechanical deficits like septal defects and valve occlusions. And they wanted a pacemaker that would be self-sufficient and could be passed through a 3 millimeter cannula that they used to go through the abdomen of the mother into the uterus and into the chest of the fetus. And so we developed such a thing. Turned out the technology was sufficient, and we came up with some rather simple designs that we could build in the laboratory and test in animals.
3 millimeter is pretty small.
Yes, it's like 1% of the volume of a typical pacemaker.
Right. And it's amazing that the pacemaker, so the kid is born with a pacemaker then.
Pardon?
Then that kid is born with a pacemaker.
So that leads to the second development, which is that the pacemaker we came up with was designed simply to get through the last month or two of fetal life so that the child could be born fully developed. At that time, they would continue to need pacing. The fetal pacemaker would almost certainly either not survive birth or not be suitable for decades of pacing going forward. So we've been working for the last few years on a leadless pacemaker that can be minimally invasively implanted on the outside surface of the heart, because a young infant is too small for an intravascular pacing lead. So that's another series of animal experiments that we just finished, which we think would be valuable for the children who undergo the fetal process, which is a very rare disorder, for other children who need pacing in the first couple of years of life when they're too small for a conventional pacemaker. And if it works well enough, perhaps for the majority of all pacemaker implants, where you're worried about lead life from the constant motion of the heart and where you want to be able to implant something minimally invasively that doesn't leave a device that could poke through the skin and need to be surgically replaced when the batteries wear out.
Right. That's phenomenal. Right. Okay, so that's about pacemakers. Let's come back to, one of the favorite topics of myself. You are also active in that, and a lot of audience of this podcast, the control of movements. So the gap between what happens in the brain and what happens, in muscles. Our understanding of how the brain controls, or if we are right saying the brain controls movements. So where are we with respect to that understanding? What's your state? Okay, so for instance, whenever on an economics podcast, there's an economist, the first question they're asked is, what's the state of the economy? So my question to you is, what's the state of our understanding of motor control for whatever motor control means?
There are parts of it that we understand quite well. But many parts of it, and particularly those that would be required for a prosthesis, that we don't understand sufficiently. In particular, we really don't understand the coordinates that the brain uses to decide on strategies for achieving movements. The brain doesn't talk directly to individual muscles. Most of its output goes to a very complex set of circuits in the spinal cord that I've spent much of my career trying to understand, as have many, many other neuroscientists. This is a field that goes back now over a century. And it's the level at which all the signals come together, both the descending commands from the brain and a vast amount of somatosensory feedback, both cutaneous input and proprioceptive, that feeds back information about the position of the limbs, the length of the muscles, the forces that they're generating, and the contact with the world that you get from your skin. To get some idea of how important that information is, and we've been involved in developing sensors to try to replace that. I often ask my students to think about how their hands didn't work on a day when their fingers were numb from the cold. That's a scenario that worked better when I was at Queen's University in Canada than now at Southern California.
Hmm.
But if you've ever had your fingers numb from the cold,
Yeah.
you'll realize that your ability to manipulate objects is almost gone.
It's gone. Yes.
And there's nothing wrong with your muscles. The muscles that operate your hand their fingers are not by and large in your hand.
Mhm.
They're up in your forearm, which is perfectly warm and functional. All the descending commands from the brain can get to those muscles. All the mechanical linkage is fine. It's not affected by the cold. It's just the absence of contact information in your fingertips that completely befuddles the brain and the spinal cord circuits to make use of the strategy that you might have spent your entire lifetime learning on how to control your fingers. Suddenly, without that sensory feedback, you're hopeless.
Yeah.
So, if when we're talking about trying to restore function, like dexterous manipulation, we have to produce not just a way to find out what the brain is trying to do, which means reading out signals in some coordinate frame that we don't yet understand, but then how to integrate those commands with sensory feedback, which means some way of recording that sensory feedback and some knowledge about how it gets integrated to produce these functional movements. I think there's a lot of roles for exercising muscles with stimulation, and that's the area that I mostly worked in. Because as a clinician, I know that many of the things that happen that are most distressing to patients with paralysis or weakness are not the loss of function, but the secondary effects, things like contractures and blood clots and pressure sores, things that cause them a great deal of
Yeah.
medical distress, morbidity, and even mortality
Yeah.
that are actually relatively easy to fix with just exercise, non-specific exercise of the muscles,
Yeah.
which is relatively simple to do with devices.
Right.
Whereas replacing complete sensory motor functions, voluntary functions, is still, I think, well beyond the state of the art.
Right. That's true. That's true. So, okay. So when you said that the spinal cord lies between our brain and between our movements, right? Between muscles. Brain does not directly interface the muscles. A lot of what we know, especially in the field of motor neuroscience, is basically correlations between movements of different body parts and brain activity. It can be direct measure of brain activity, using single electrodes, in monkeys, or it could be, brain imaging, or it could be, for instance, brain stimulation like TMS. So when we have ignored the interface, but we know the output and we know, something happening in the brain. So what is that knowledge useful for? Ignoring that interface and ignoring that translation.
As a clinician, you have to think about what it is you're trying to fix. Uh,
Hmm.
the as a scientist,
Yeah.
we have a tendency to try to identify what the brain does by looking for correlations between whatever measure of neural activity we have,
Yeah.
imaging or recordings, EEG, microelectrodes, whatever it is. We correlate that with observable aspects of the behavior.
Yeah.
And then we say something which is very misleading, which
Yes.
is that part of the brain must encode the
Yeah.
variable with which it is correlated.
Yeah. Or represent in a different facet. Yeah.
The problem is that by its nature, a sensory motor plant has everything connected together.
Yes.
So everything winds up correlated with everything else. You could say that the muscle encodes the activity, the brain encodes the activity of a muscle, except if the muscle is activated, it's going to cause changes to the position of the joints. So there will, of course, also be a correlation of that brain activity with the position of the limb or the velocity of the limb or the
Yeah.
acceleration of the limb, which is what muscles actually generate. Um, and then if the limb makes contact with something, there'll be sensory information coming from the skin. So when you just look for correlations, you will always find them. They might reflect, and not only do they get correlated with all the things that you can measure, but they may be causative or they may be responsive. So when you see a
Mhm.
signal correlated with something, you could say, well, it must be commanding that change in the state of the system. But because you have all the sensory information coming back, it could just be reading out the sensory information coming from the state of the system that some other part of the brain caused to have to happen.
Right.
So we know very little when we look at neural activity in terms of where it is in the control loop.
Hmm.
And if the control loop is broken, which is what happens in most of these pathologies, uh, we don't really know what to do with the signals that we're recording. We don't know what they're going to look like, and
Right.
we don't know if we use them to make some artificial change to the system, like stimulating or running a robot or whatever it is. We really don't know how those signals are going to behave in the pathological condition because we don't know how they arose in the natural condition.
So, but that's a dominant framework of motor control, right? If we,
Sad
it,
but true.
right, like majority. But at the same time, all of that has been funded through the idea that that will lead to, some kind of benefit with respect to health, right? The implication has always been historically that it will improve human condition. So where have we gone wrong there? Like, is the other approach not possible to actually, work in the lab? Or is it that, like, how did this culture, kind of solidified itself over time? Because now, uh, I mean, if I'm trying to recall, I can't recall many of the studies which are not correlative in nature.
So science, the benefits of basic research and its relationship to clinical advances is very indirect and often very tenuous.
Hmm.
We don't really know which bit of knowledge breakthrough will be necessary or sufficient to achieve any clinical result. And often the science that winds up leading to a clinical advance only does so after decades and many other bits of science and technology come together. So I wouldn't denigrate doing experiments that are still far enough from a practical application that you can't see the link because that's most of
Hmm.
science.
Yeah.
Uh, where we have to be cautious is in overinterpreting the results of experiments. Any experimental data is, assuming the experiment's well designed and well reported, likely to be valuable and grist for someone's mill.
Hmm.
Uh, so finding these correlations is not a useless exercise. It's overinterpreting them and assuming that because you've observed them that they mean something for which there might be plenty of other explanations. That's where we get ourselves in trouble.
Hmm. Okay, I see. So, okay, so two questions now related. So one is, how can we go actually from how can we move away from causal from correlation to actually understanding the loop with a physiological horizon in mind? Uh, and second, where can we actually use this correlation? Because everything is correlated with each other. So what kind of information they actually produce? So when we say that we have understood motor control, so I mean, I won't name, but there have been articles with the titles like the neural basis of certain things, right? And you see that those kind of articles like 2 decades back, then a decade back, and you can see them in probably last few years, right? The neural basis of that. So the idea is that we have understood the neural basis. So, one, how do we move away, keeping the horizon of physiological interpretability as well as applications? And second, when we say that we have understood the neural control, what exactly would be the benchmark when we would say that we have actually understood? So, do we have that horizon of understanding clear to us, or we don't even know that horizon, that what understanding actually looks like?
So I spent the first 30 years of my career doing experimental neuroscience and observing activity patterns in mostly single neurons in behaving animals, both sensory and motor, and trying to make sense of it by looking at how those activity patterns related to the functions that I was observing in these naturally behaving animals. And that's a situation, and all of that was work in the spinal cord and peripheral nervous system, where the relationships are relatively easy to work out and where the circuits are relatively well known. But even that turns out to be actually quite complex and contentious. The last 25 years of my career, I've done much less experimental neuroscience and much more modeling, because I think from the experimental neuroscience, we get ideas about how the thing might function. But until we construct quantitative models of the whole system that embody such theories or how they work, we don't really know if those theories are going to work. And we don't really know how to design experiments to test them. That is, if you have a working model of the system according to your theory, then you can say, if I make this intervention here, then this perhaps unexpected result should occur. Now I can do that experiment in animals. So developing larger models, which fortunately is now more practical given the advances of computing, is an essential step to testing theories. But the question then becomes, how do you bound those models? Because the nervous system is very large, very complex,
Yeah.
and there are a very large number of parts that are involved in any particular behavior. And one of the pitfalls that we all fall into, whether we're experimentalists or modelers, is we think that the particular subset of the nervous system that we are recording from or making models of must be responsible for the behavior. While, if it weren't, why would I be wasting all this time studying it? It's a sort of circular argument, isn't it?
Mhm.
And we have to constantly ask ourselves, which parts did I leave out? How are they connected to the parts I modeled? And what might they be doing to change the way the system works that's not captured by my model? That actually used to be easier to do, because in the early days of the neuroscience that I was involved in, the field was so small that when you went to a conference, you got to hear about all those other parts.
Yeah.
So, the first neuroscience Society for Neuroscience meeting I went to had 500 people. The last one was 35,000. So in a conference with 500 people, the couple of hundred people who were working on the entire sensorimotor nervous system would hear about all the different parts. In the same session, you'd have people talking about spinal cord and cortex and cerebellum and superior colliculus and eye movements and legs and animals and humans. You got the sense of the scope of all of it. You realized what you were working on and something about the other parts that existed and who might be able to tell you about them. These days, there's so much information about each one of those subsystems that it's difficult to even pay attention to the other subsystems. But they're out there, and anybody who's modeling a system has to be constantly asking themselves, what am I leaving out, and how might the parts that I'm leaving out change the behavior of the system that I think I'm modeling?
An example?
So for the last 6 or 8 years, I've been working in a collaboration with Tim Carroll at the University of Queensland in Australia on a notion that's been floating around for about 20 years, that the uh, that an old part of the midbrain called the tectum, which in mammals consists of the superior and inferior colliculi, it's might have some very important roles in sensory motor control of the whole body. As it turns out, superior colliculus was extensively researched, specifically with regard to oculomotor movements. That's a subsystem of sensory motor control that we know a great deal about, and for which the superior colliculus and some brainstem structures to which it projects are responsible for some very sophisticated dynamics of target acquisition and uh, and planning and control of muscle activation. It was largely overlooked that the superior colliculus uh, projects via the reticular formation in the midbrain and brainstem to the spinal cord. It's been identified that those projections are also responsible for gaze control involving the head, because it's not just the eyes that move, it's the head that moves also. And now it's become clear that it has a potentially important role in the control of both the upper and lower extremities. And the question becomes, well, it's a part of the system that people studying limb movement have completely ignored. And you have to ask, if you have this sophisticated system, remember in the lower vertebrates that have virtually no cerebral cortex, the tectum is responsible for all of their sensory motor behavior. The tectum and the reticular formation are able to do the very sophisticated things that are involved in animals detecting and acquiring prey. So if you have this system, and it's still present in the mammals, including humans, that's capable of doing these very fast, very accurate transforms from sensory stimuli to motor behavior, maybe the late developing motor cortex that we spend so much time studying is taking advantage of all that computational machinery that's been largely overlooked. And so the experiments that I've been involved in have started to reveal how that system contributes to rapid reaching movements to targets, and perhaps what its computational capabilities could be utilized by the cerebral cortex, which would really transform the job that we think the cortex has to perform. If it's essentially subcontracting these transforms to subcortical structures, then the whole nature of the signals and the processing that's going on in the cerebral cortex is different from the functions that we assume, from the functions that we correlate it with when we say, well, it's encoding the activity of muscles. It may be encoding very high-level things that are transformed into the activity of muscles by completely other parts of the brain.
Yeah. So that would be basically a reinterpretation of a lot of existing data.
Yeah. And the implications, if you want to get to clinical applications, of what signals we actually find in the cerebral cortex that might be useful as commands for prosthetic devices or assistive limb prostheses and robots.
Right. So one of the things points or feature of your the way you describe the motor system and motor control. I think you come from a very realistic framework or realism framework, to it, where you have an end in sight in terms of the physiology. So basically, you have physiological terms across the whole description. Now, when we look at a lot of motor control now, we also talk about motor control from an algorithmic standpoint, where we are not interested in the physiology. We are interested in only the algorithms. going back to the description, the tri-level description of behavior by David Marr, right? Uh, from algorithmic to implementation to biological, plausibility of that implementation. So where do you see, like, so what's your opinion of that framework? Like, what is the exact use of that framework for a motor control scientist or a neurophysiologist? And do we even need that framework, or we can just talk physiology and the flow of the current or the flow of whatever we measure in terms of physiology and not invoke algorithm or implementation at all?
I think that David Marr's formulation of this is really very important to consider. The top level theory of computation is essentially the block diagram that engineers use to describe a complex system.
Yeah.
How the functionality that you can observe is broken up into computational elements that are done in different places by different structures with running different algorithms. If you don't have a valid theory of computation, then anything you do to interpret what's going on in one of those boxes is going to be wrong, because if the boxes actually subsume the activity from multiple, you simply don't have a representation of what to look for in the box unless you know how all the different aspects come together. And that's the theory of computation, how you divide the problem up. Because we assume that the different anatomical parts of the nervous system are doing different parts of the problem. We could guess how the problem might be divided up by engineers. The engineers who design robots have to do that. They have to say, we're going to compute this at this level, we're going to compute this at this level, and that's how the system goes together. But the fact that you can do it that way doesn't mean it's the only way it can be done.
Yeah.
Nor does it mean that any of the different ways it could be done, any one particular one of those, is the way it's actually divided in the nervous system. So everything I said earlier about putting together a systems diagram of and modeling your theory for how it works involves assumptions about what's in those boxes and what's in the other boxes that you have chosen to ignore, which might be changing the way the system works. So I think the theory of computation is essential. It's one that is often honored in the breach. That is, that things get left out just because we don't know about them, we don't have a model sufficient for them. They seem like an unnecessary complication. You don't have the computational capability to include those models. So models are always going to be leaving things out, but we have to have a theory of computation that justifies that. That says
And
for this particular function, we have
yeah,
a reason why we think we can leave those other parts out.
this theory for computation, would it be axioms or would it be actually, should it stand to falsifiability?
It has to lead to falsifiable hypotheses. It's interesting that in David Marr's book Vision, 1982, I believe,
Yeah.
when he proposed this hierarchy, this three-level hierarchy, he did so while presenting a theory of computation for 3D vision,
Mhm.
which turned out to be wrong.
Yeah.
But the important contribution was being able to define the theory for the complete functioning of the system in such a way that the experiments could prove that the proposed theory for how it works was right or wrong, as opposed to simply letting researchers muck about in the system, recording data, reporting correlations, and having no idea whether they're actually sufficient to test a theory of computation.
Right. Which is a very pervasive problem in modern neuroscience right now.
Yeah, we collect a great deal of data without knowing if it is, I mean, as I said earlier, all data are useful and might be grist for someone's mill. But we would like to think that the data we're collecting now is immediately important to some
Yeah,
problem.
right.
And without a theory of computation, we don't know that.
Okay, so now coming to the second level in David Marr's hierarchy of investigation. So second is the implementation, right? So what do you mean like with that, and where do you see in motor control? What are the questions we have to ask at that level?
So there's an interesting constraint
Yeah.
on the nerve on the biological system, and this is a tension. Let me back off. There's a tension between the engineering and science.
Hmm.
Engineers get to invent a solution, and if that solution solves the problem at hand, then it's an acceptable solution. That's it. And there may be a large number of different inventions that could accomplish the task, and they don't care which one it is as long as it solves the problem. They might
Yes.
compare which one's more efficient, which one's faster, which one uses less energy, whatever. But any solution to the problem, if the problem is well stated, is acceptable. Scientists have a very different problem. Scientists have to just don't get to invent any solution to a problem. Scientists have to discover the one solution to the problem that Mother Nature invented out of a very large number of possibilities to solve each individual problem at hand. And so you need some constraints to tell you how to compare your different theories. I mean, we don't get to ask Mother Nature directly what the solution is. What we do is look at a lot of data and make
Yeah.
guesses about how it might be done. And we might have lots of different guesses. And so now we want to say, well, how do we compare
Hmm.
those different ones and get a sense, an intuition for which one is likely to be closer to the one truth that's actually present in the biological system?
Yeah.
So what we need all the constraints we can apply to it. And some of those constraints involve the capabilities of neurons to process signals. There are some things neurons are good at and some things that neurons are bad at. It's baked into the biophysics of how cells use action potentials and synapses to transmit and compute. So it's a constraint that whatever you think is happening in one of these little boxes has to be something that neurons are suitable for. You have to have an algorithm that can be embodied in neurons, or what we call neuromorphic computing. You also have to worry about, and this is a constraint that has been given much less attention than it deserves. You have to worry about how the system evolves, both phylogenetically and ontogenetically. What do I
Yeah.
mean by that? Well, first of all, you get a lot of hints about how the nervous system could be operating by looking at simpler nervous systems and realizing that things that were put in place that are still present and that other things had to be involved on top of them. And that in many cases, there's a famous paper by Lloyd Partridge from the 1980s called Evolution is Not Engineering, which is that the structures that you find in an advanced organism are not necessarily the best solution to the problems that the advanced organism has to solve. They may be accommodations of much simpler mechanisms that were developed at a time when the requirements were very different.
Yeah.
A classic example of this is the mammalian eye, which is an engineering disaster
Yes.
because the photoreceptors are on the wrong surface of the retina, which didn't matter when resolution was not a problem, when it was just a vague extension of the brain trying to detect light. but which becomes a nightmare of highly specialized structures designed to overcome the fact that all the crap on the outside of the photoreceptors is screwing up the transmission of light, and you put in all these corrective circuits to deal with that problem because you couldn't flip the retina around once you got along the phylogenetic path of building retinas in the first place. So a large part of the complexity and function of the retina is not something an engineer would have done from scratch. But if we think about how retinas evolved, we can start to understand what the structure and function is of the retina and the visual pathways based on the constraints imposed by evolution. We also have to consider the constraints imposed by ontogeny, that is the development of individuals.
Yeah.
What's the information and process whereby this vastly complicated system gets itself wired up?
Mm-hmm.
And that's something that I've been writing on for the last few years.
That's like magic.
Well, we often, for a long time, we had to assume it was magic.
Mm-hmm.
Then genetics came along and started identifying all these different markers, genetic markers that are expressed in the different neurons, and started to make the assumption that that's what the DNA specifies, that all the connections in our nervous system are specified magically according to the vast number of genes that are available to say what should get connected to what. And that turns out to be a bad idea that was actually recognized in 1896, because if you had genetic programming of both the physical plant, musculoskeletal system,
Yeah.
and its controller, you have an evolutionary dead end.
Right.
So I don't know if you want to continue on this vein. It's
Of course,
an
of course,
interesting topic.
of course.
So let's imagine that we have fish in the sea and their nervous systems to control their swimming, their fins, has been genetically specified to do that really well through hundreds of millions of years of evolutionary competition. Now, one day, a fish develops a mutant fin that's not terribly good for swimming, but is able to support the fish's weight on land. There are no land animals. First fish is going to come out of the sea and discover this vast niche that could be occupied by anything that can survive on land. And it's got a mutant fin that might actually be useful in that direction.
Yeah.
It's still extremely crude, and it's going to take hundreds of millions of years of further development to turn into limbs, but it's the start. But for that fish's mutation to survive and be further mutated by its offspring, that fish has to survive. If its nervous system is entirely programmed to be, say, the optimal controller of fins swimming in the ocean,
Yeah.
it's dead.
It's that, yes.
End of evolution.
Right.
So Baldwin, the scientist thinking about this problem, came up with the notion that the controller, the nervous system, has to self-organize. It has to figure out what the properties are of the physical plant it's trying to control in the individual and wire itself up to take the best advantage it can of whatever it finds.
Yeah.
And that is a very powerful constraint on the kinds of nervous systems that you can evolve. You still have lots of genetic signals telling you things that are related to how it's going to be wired up, but they're not the things that actually wire it up.
Right.
Those genetic pathways are telling you maybe what kinds of neurons you'll develop
Yeah.
and when, particularly the timing of their maturation, when they start to make connections,
Mhm.
and maybe some controllers for spontaneous activity,
Yeah.
which we know occurs in fetal organisms, and that milieu in which the thing self-organizes is what allows you to overcome this dilemma so that mutations can get immediately used by the extant nervous system. And it provides a very powerful constraint on the adult systems that we actually get to observe.
So by that token, we we should actually bring it even at the level of, lifetime or smaller time scales of learning. So for instance, if I'm driving a car, right, I'm using a steering to drive a car, which is a very different kind of mode of locomotion, where or I'm interacting with the environment, very different kind of contacts. Likewise, when I'm having a prosthesis or I'm using a tool or a wiper to, wipe a screen, again, I'm having a very different kind of motion, right? And the controller is self-organizing. So should we extend that even to our our interactions mediated by objects with the environment?
So we spent the first few years of life making tens of millions of trial and error behaviors to learn a repertoire of sensorimotor skills.
Yeah.
We have some very limited pre-programmed reflexive pathways built in. We have some things that we acquire probably in utero by this spontaneous self-organization. And then we spend the first few years of childhood acquiring these behaviors, a repertoire of behaviors, during which time the higher parts of the brain, cerebral cortex, are discovering the properties of the rest of the system that have emerged during this developmental phase. How does it do that? The cortex sends signals out. The neurons in the cortex have no way of knowing
Yeah.
where their outputs go. A pyramidal cell in the motor cortex sends signals to many different interneurons and many different muscle groups in the spinal cord, a whole bunch of signals to the reticular formation and to other subcortical motor pathways. It doesn't know where those signals are going. It gets a lot of inputs, synaptic inputs, doesn't know whether those synaptic inputs come from other cortical cells, other cortical regions, other subcortical structures, or they represent afferent information, sensory feedback coming in
Yeah.
from the thalamus. So some simple rule that causes them to change their input-output properties has to result through the emergent interaction of all of these circuits
Mhm.
in useful behaviors.
Right.
So we need some mechanism that can try a bunch of different things. And you see this in infants from the very
Mhm.
time they're born and start making what appear to be relatively random movements and gradually acquire more sophisticated movements, presumably because they have some mechanism that can both motivate these trial and error experiments of sending out new signals and can look at the results and decide whether or not to remember those patterns
Yes.
of output. it seems likely that the particular representation of behaviors that each of us winds up with is very different at the neural level because we may have stumbled on very different ways to accomplish what appear to be the same tasks. And that may mean that as adults, even though we all appear to be performing normally, we may find it difficult or easy to learn new tasks because the new task might be similar or not to something we already know.
Yeah.
Let me give an example. And I was at a motor control meeting, must be in the 80s, in a picturesque place in Ohio. One of the hot new things that people were talking about was windsurfing.
Mhm.
So here are a bunch of sensorimotor neuroscientists, all interested in the acquisition of skills, all reasonably healthy and fit and normal in their function, but essentially none of them have ever tried windsurfing before. So we all go out to the lake and we all get these windsurfers, and you look at the situation 2 hours later, and what you find is that some of these folks are up on their windsurfers skimming across the lake with, abandon.
Yeah.
And others of these folks, after 2 hours, are still falling down in the water every time they get up. These people were all normal individuals with no apparent differences in their athletic capabilities. Why did some find it easy to acquire this rather unusual pattern of
Yeah.
sensorimotor coordination that you need for windsurfing, and others found it almost impossible? I suspect because the ways that they were accomplishing the tasks that we normally think of as normal tasks that everybody does the same way were in fact internally represented in their brains in very different ways. And some of those ways were close to good enough solutions for windsurfing, in which case it
Yeah.
was easy for them to find it. And some of those ways, which are equally good in their natural performance of conventional tasks, were very far from what you needed to windsurf. And so in 2 hours, they never got out of what turned out to be a bad local minimum in engineering parlance.
So would you equate these, learnability to motor primitives?
No.
Why not?
Well, all right, let's talk about what we mean by motor primitives.
Yeah. It's been a controversial topic for the audience, right? People
Yes.
believe in motor primitives or not, or what exactly they count, and there are ad hoc or post hoc explanations, generally
So it
speaking.
depends on what you mean by motor primitives
Yeah.
or synergies, another
Yes.
term, a related concept. The notion was originally put forward by Bernstein and others, particularly in the alpha and gamma hypotheses, that the nervous system has available to it only a limited subset of possible circuits or possible ways of coordinating muscles or movements or joints or whatever.
Yeah.
That not all things that are anatomically possible are actually neurophysiologically possible. And that this was a solution to what's called the redundancy problem, where because the musculoskeletal system is said to be overcomplete, itself, I think, a misnomer we could talk about, that the nervous system would simply be too confused by being able to use every possible combination. And so it evolved to have only limited circuits, which would be useful circuits, which the system would then use to quickly find solutions to problems. Now, if you think that the primitives or the synergies are sort of hardwired into the system, then you're making a strong hypothesis about the actual computational structure of the nervous system.
Yes. And hardwiring the control.
Yeah, and limiting its performance.
Yeah.
Now, what I was talking about was a system in which pretty much all things are possible, but as a result of experience, you develop a repertoire of capabilities. And when you need to do something new, you work from the existing repertoire to develop an extended repertoire, which is modifications of the existing repertoire.
Mm.
What that means is that in any given individual, you're likely to observe that motor behavior is habitual.
Yes.
That is, you tend to reuse stuff that's good enough to do what you want to do and to make your changes incrementally from there. But that, those habits that you rely on as the building blocks for further learning are not hardwired. They didn't exist in your genes or in the way the nervous system had to be. And with sufficient practice, learning, they can be overcome. You can get out of habits. In fact, that's what coaches and physical therapists have to do with people who find themselves in dysfunctional states, get them into patterns that they don't seem to have available to them, but they could learn to have available to them.
Yeah.
So we could say that in a given individual, you have habits. Those habits tend to limit function, at least in the short term. Are those primitives? Are those synergies? No, they're just tendencies. So it's very important to distinguish what you mean by primitives or synergies as to whether you're implying that they are inherent to the organism, inherent to the species,
Yeah.
that they're part of our baggage and on an evolutionary level, or that they are simply an emergent, they're just a word describing the emergence of habitual performance,
Mm-hmm.
which is not a very exciting concept, is it?
It's not, yeah.
We talk about synergies and primitives as if there's some actual wonderful fact that we can do experiments on to reveal and discuss and publish papers about.
Yeah.
But if they're just the statements that people tend to keep doing the same thing that's worked for them before, it doesn't get you very far in science.
Also, it's not clear how much of these, I think we have not done control experiments to figure out how much of what we can understand is artifacts of our, because we are getting increasingly mathematically elegant. So how much of what we obtain is mathematical elegance versus actual physiology or actual, understanding? Because certain mathematics is kind of a lens, right? Every lens bias you to look at certain things. And we need a great understanding of what exactly that lens is biased to look for. So I think we have gone a little astray on synergy because of those mathematical elegance as well.
Well, it turns out that there's a very nice set of analytical tools from which you can extract synergies.
Yes.
So you can look at a very complex data set and observe and summarize it with some fidelity
Yeah.
in a very smaller number of dimensions,
Right.
which we call synergies.
Right.
But the fact that we can summarize a data set in a simpler low-dimensional term doesn't mean that that was actually done in the nervous system.
Yes.
And we have to ask, what is it that limits the number of synergies that we actually extract with these methods? One of them is, how much of the data do we actually have to account for? How accurate does the model have to be? This
Right.
is what we call variance accounted for,
Hmm.
which, when people get to 90% of the details of the curves of activity in all those muscles, they say, well, okay, I needed 4 synergies to account for 90% of the variance in all of these behaviors. And so I'm happy that's the system. Well, what about the other 10%? Does it matter?
Yeah.
Uh,
So
and
the
there
yeah.
and there've been mathematical simulations of what happens if
Mhm.
you leave 10% of the EMG unaccounted for, and it produces disastrous performance.
Yeah, exactly. The other question is that 10% the death and of the importance of death and life, or is
Yeah.
that 10% has nothing to do with, because reaching 90% is fine, right? But that 1%, that last 1% can be the key to dexterity, or to be able to reduce a fall, for instance, avoid a fall, or any other variable which is important to live.
And that leads to the second problem,
Yeah.
which is, how much data did you have
Hmm.
to account for? So
Right.
an example I included in my
Hmm.
paper on learning to use muscles is one that I know well as a piano player. We know that the piano has 88 degrees of freedom. It's got 88
Hmm.
keys.
Hmm.
Okay. But suppose you didn't know that.
Right.
Suppose you just listen to compositions played on the piano. And from the patterns of the notes that you heard, you would decide on how many degrees of freedom the instrument had.
Yeah.
So if I listened to a whole bunch of compositions by J.S. Bach in one key, I would have a relatively small number
Hmm.
of synergies that would account for Bach's music.
Yes.
If I were to extract the synergies by listening to the songs in the 24 pieces in the Well-Tempered Clavichord, which are all in different keys, that series of compositions was actually written to prove that you could tune a piano so that it
Hmm.
would work well in the 24 different possible major and minor keys. So you would have to include a much larger number of synergies.
Yeah.
Now, if you were to listen to the piano and you were playing Bach and Beethoven and Chopin and Stravinsky, you would wind up with a much larger number of synergies.
Right.
Um, in the end, you would probably need at least 88 synergies, which happens to be the number of degrees of freedom that the piano is limited to.
Right.
So,
Right. So this is where when I started, I asked you, so when we say that we want to understand something, okay, coming back to the epistemology of science, when we say that I want to understand the system, and I myself struggle with this, because, as an undergrad, I used to make fun of research papers that most of the papers ended up with the future directions. And me and my friends were looking for a paper which says, like, this is understood now. Okay. Now we understand the whole idea of, the island of knowledge, and the more information you have, the more ignorance spreads, exponentially perhaps. But still, we still need to have an insight that we want to understand it. So when we come from a realistic framework that we want to understand it for some, let's say, repairing movement, we have an end in sight. But as a basic scientist, when we go to study motor control, we generally don't have an end in sight. So what an end in sight of understanding look like? Because
well,
if we, yeah,
you 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 left out a third way, which is the most important way.
yeah,
You suggested that the future work, the discussion conclusions might say that more work is needed along these lines.
mhm,
Or they might say that we're done here and we've learned, everything we need to know along this line. The third conclusion, and perhaps the most valuable one, is we've been doing this for a while and it's a dead end. This isn't going to lead to the truth.
Hmm.
And if we think about it, that's really what science is all about. Remember, when we test hypotheses,
Yeah,
we don't prove they're right.
yeah,
We only fail to prove they're wrong.
right,
What that means is that all we can do in science is reject things.
right.
I like to, an analogy I like is imagine you've got a large block of granite and you want to make a beautiful statue. You, the process of sculpture is removing the bits of rock that don't belong in the sculpture.
That's true.
So if in science we're trying to discover reality, we're constantly looking at bits and pieces that don't seem to fit with that reality and realizing you have to remove them. And sometimes we find that the processes we're using aren't sufficient. So one of the conclusions, valid and useful conclusions of a whole line of experimentation could well be that this, we've gotten what we can out of this, and it isn't going to get to the truth.
But that the
Doing more of it isn't
sociology,
useful,
yeah,
and we need to do something. And it's not that we got to the truth.
yeah.
It's not that we need to do more of it to get to the truth. It's needs that we need to do something else.
But that the sociology of science doesn't allow so much, right? Because
Ah,
then, yeah,
of course. We generally find it easy to get funded to do more of what we're already doing and already productive in.
yeah,
And as soon as you say, I need to do something completely different, uh, that means I'm going to need a whole bunch of expensive new equipment, whether it's computers for modeling or software or new apparatus for the experiments or new animals and species. I'm going to need a lot of startup.
hmm,
And when I go to apply for that, it, I have no track record showing that I've been productive at it, either because it's completely new and nobody knows if it's going to work, or it involves going into a field for which I have no track record. So, after spending 10 or 20 years comfortably getting grants from study sections that know my work and have seen me at meetings and
yeah,
review my papers and all that, I suddenly find myself in a completely new study section with people who look and say, who the hell is this guy?
hmm.
Not this is somebody who's published hundreds of papers and has a certain, that doesn't count anymore.
That's
So
true.
yes, the sociology of science makes it very difficult
Yeah,
to come to what I think is often the valid and correct conclusion, which is we need to do something else.
right. Okay, so I'm in motor control, like one of the most topics which interest me most is the control of movement, perception and action, more at the, meta level, but of course the details. So, so now we have in motor control, we have, starting from journals to individuals to, study panels, we have these demarcations where there are abstractions of internal models. We have physiologists and we have, the people at the, so basically Marr's three-level divide is there, is basically has divided. Now, even to complete Marr's program, we need to be able to, draw a trace between the three, right? So if we have a metaphor or an algorithm, we have to bring that metaphor to the implementation level. And if it cannot be translated, we have to abandon it and improvise it or abandon it and look for a new computational, map. Likewise, we have to bring to the level of physiology, right? And if we know that something is not biologically tenable to begin with, that means that's not how the body works, right? It's our fantasy. So now where are we in motor control? Where do you think would be the most fruitful avenues moving forward? So for instance, when we talked about the sociology, for a young person who is starting a faculty position with a startup, it's a great opportunity to start something fresh. right? That's what startup is for, right? Or when you move positions, for instance, or when you have an endowed position, uh, you know, because then you are not liable, to continue the inertia. So where do you think the most fruitful pieces are, or what are some of the puzzles where which can bring together now to have the most fruitful program moving forward in motor control?
Well, nobody really gets to start fresh. That's a bit of the baggage of the way we organize our profession. The laboratory that you find yourself in as a graduate student is often one that you fall into either because they happen to have an opening or you happen to have an interest, but you make that decision either someone else makes it for you or you make it at a time when you really have essentially no useful information
Hmm.
about how promising this field is. Students, in fact, often make what I call the worst possible decision, which is to go into something that's currently hot.
Yeah.
Now, it's going to be 10 years before a starting graduate student becomes a really effective professional contributor to a field. You know, half of that spent getting your PhD and another half in postdocs and the first beginnings of an assistant professorship and being an independent researcher. So it'll be 10 years before you're really and it takes about that long just to get efficient at being able to read the literature, extract and for the methods and get familiar with the statistics and how you write papers and all that stuff. So if you go into a field that's currently hot, you're 10 years behind the curve.
Hmm.
And it's a pretty good bet that any field that's currently hot is going to be pretty well played out and derivative in 10 years. So that's really not a very promising strategy. You need to go into something that's not hot. Well, you know, you're hoping that in 10 years it'll be hot about the time you're in a position to contribute to it. I don't know any answer to that problem, but it's certainly worth thinking about things that look like not that they've already been highly productive, everyone's talking about them, but that perhaps they're on the cusp in terms of science and methodology coming together in the next 10 years to do really great things. So that's one bit of advice. But once you get on that track, your future opportunities are heavily constrained by what you've produced and who you know.
Right.
So it's not like you really get freedom. And if you finish your PhD on a topic, it's likely that your postdoc will certainly be something related to it because when people hire postdocs, they're looking for people who have at least methodological expertise that can be immediately applied in their laboratory. Hopefully, you get a postdoc in something that provides you with a broadening of your methodological skills, but it's certainly going to be in an area that's closely related to what you did your PhD on. And then when you go to apply for an assistant professorship, they're going to want to know what you plan to do and how you plan to get it funded and what startup you're going to need and what equipment and what sort of courses you're going to be able to teach. And if those are not closely related to what you're already known for,
Yeah.
probably not going to get the job. So yeah, there's no easy time at which to look at something and say, this, this is not what I want to continue doing.
Hmm.
The trick, I think, is to be constantly thinking about whatever it is I'm doing now might either be played out or a dead end in the next few years. So while I'm doing this, and while I'm able to keep my lab funded and pay for the rent for myself, I should start looking for other things to do on the side, other collaborations, other things to read, ways to bootleg projects to try something new that doesn't require a large amount of external funding and approval by study sections, so that you can anticipate that you are going to have to phase into something completely new, start to get the credentials into something completely new, and then make that transition before it becomes so compelling that you're stuck.
Right. So how have you navigated like in your so you have a very consistent record of productivity, both on the front of basic science as well as in the front of, innovations in biomedical sciences. So have you been constantly funded in your career, or like how did you navigate the whole landscape?
So I've been able to, if you mean by consistently funded, always having support for the
Yeah.
research I want to do, yes. If you mean consistently funded, uh, having long-term support on a particular problem or a particular methodology, then no. I always tell my students I have a short attention span. I can't keep doing the same thing for more than 5 or 10 years.
Yeah.
So I'm always thinking that what I'm doing now is likely to come to some kind of end. And
Mhm.
I'm always looking at other opportunities along the way to try things that are either, related to what I'm doing now, but different, or completely new, fields that I find I'm intrigued by,
Yeah.
and I can cut myself enough slack to get involved in while I'm still working on the thing that everybody thinks is great. Now that has an interesting arc to one's career and reputation, which is that if the about the time that what you've been working on and what's been published starts to be recognized and cited by your peers, you might no longer be in the field,
That's my problem. Yeah, that's been my problem.
and you're working in some other field.
Mhm.
So, it's very comfortable to stay in a field where you're known. As we said,
Yeah.
study sections know you, you know, reviewers on journals know you. You get invited to symposia at conferences, and everybody talks to you because you're the grand old man of this field.
Mm.
Except what it gets less and less rewarding in the sense that you
Yeah.
you don't think it's going to go in the important future directions. And if you move in 10 years to a new field, about the time that you could have been resting on your laurels, you're back having to justify your existence to a bunch of people who don't know you. Um, it's not easy, but I personally think it's more rewarding. And reinventing yourself is the term that people use now for these kinds of career shifts, which I think are crucial in science, and certainly for myself have been very rewarding.
Right. No, that makes sense. And you see, it's very apparent when you look at people's vita, the two kind of, footprints. Uh, okay.
By the way,
So
working
yeah.
in if you put any store by the H index,
Hmm.
this quantitative way of trying to compare scientists' careers, it turns out, and I only realized this retrospect, that if you publish a relatively small number of significant papers in a field and get the hell out and work on something else,
Yeah.
it actually does wonders for your H index, because people will reference the few papers that you did in the field, assuming they were significant work, and
Hmm.
they'll continue to reference that for a very long time. But if you have 5 significant papers in a field or 50 significant papers in the field, people are still going to reference 5 of them.
Yeah. So basically, so you're saying
And
that
and and
you're
then if you work in another field and produce 5 significant papers, then all of those
uh,
people are going to reference those 5 papers. So
right.
most of your output winds up being highly cited because there
Hmm.
isn't that much of it in any field. It's
So
kind of
so
an odd way of gaming the
it's
system.
Or you can say it's more like a low dimensional signature, generally speaking of most activities, right? Like 80% of results are coming from 20% of the efforts, and we are talking about those significant, works mostly.
Yeah.
Uh,
Now, the trick, of course, is that you have to make sure that the whatever it is you're doing is significant enough
yeah,
that people will, and that generally means being original enough,
right,
um, that people will feel compelled to cite it long after you cease to be a force in the field, right? So,
right,
you know,
right.
are people citing papers because they see them as foundational to the science, or are they citing them because they need to suck up to the authors that they're meeting at conferences? And
I
since,
completely agree with you, and I won't name, but there are scientists I respect, who have completely left the field for like decades now, but they wrote some of the legend articles. And you have people, again, I won't name, but I'm currently, writing a lot of articles on is basically, a citation. I mean, to be honest, like I personally don't think of citations. I think those are post hoc, as a young person, those are post hoc outcomes of good work or bad work rather than something which you keep in mind while working.
yeah,
So,
I mean, I mention this
yeah,
as a fringe benefit of taking the
yeah,
risks of reinventing yourself, not as a reason to do so.
yes, yes, I
The
agree.
important thing is to look at any potential new opportunity in the sense of
Hmm.
where you think it'll go and how interested it is and how well it fits with your skills and capabilities. There's some kinds of science and technology that I'm comfortable with, and others that I'll never be comfortable with. And so I need to work on problems that are suitable to my strengths and my interests, and that I could really engage in rather than doing it as a careerist exercise to
Yeah,
advance my promotions.
of course, yeah. Okay, so now a couple of more questions, before I let you go today, which is one of the thing is the change in landscape of research. So you almost what was going on half a century back from now, right? In terms of research, how the research environment looked like. Now, I grew up reading books of that era, I mean, a little before you, starting with the Manhattan Project. Okay, and I have a lot of fantasies of how science used to be during those times and watching movies, from that era. Uh, Now we have science at a very different level. We are asking a lot more advanced question. We have grown a lot in terms of what machines we have, what equipments we use in labs. The whole scientific ecosystem also has changed, starting from the time scale at which we can share our results. There's almost no lag between when the knowledge is produced and the knowledge is consumed, which used to be a lot, as well as the amount of knowledge which is there to consume. Uh, to competition in terms of getting funding and things like that. So. Do you consider yourself fortunate enough that you were young in that era, or you would like to be given a chance to be young researchers in the current, the way science works right now?
Yeah, there are obviously large advantages and disadvantages to both eras. The fact that our technology is so much more sophisticated and so much more rapidly evolving means that things that used to take a very long time uh, can go much faster. And that's a great advantage. Uh, the sheer scope of science, how much is being done in parallel, how easy it is to find that
Hmm.
using the tools that we have now, things like Google Scholar, um, are, that's a tremendous advantage to being in the now. What I find discouraging about the present compared to science as was 50 years ago when I was getting into it is that along with that growth in scale, we've had a cultural shift. 50 years ago, most people who were doing science did it out of love. It wasn't really a career that was seen as
Yeah.
particularly desirable or affluent. It was a calling, and people did it because they really were interested in the science.
Hmm.
And there were a relatively small number of institutions at which the highest levels of science were done, and they were filled with a high concentration of really bright people. I would guess that, in the last 50 years, we've grown the scientific establishment in my field maybe 100-fold. And the question is, well, where did that growth come from? How is it distributed?
Mhm.
Did everything scale up in a congruent way? Or have we changed the density and distribution of really high quality science in a way that's problematic? I would guess that while we've scaled up the amount of science being done 100-fold, we've probably only scaled up the amount of really high quality science 10-fold, which means that the density
Yeah, it's reduced.
of great scientists has greatly decreased.
Yeah.
And it's further been decreased because the number of institutions that scientists are working on now has greatly expanded.
Yes.
So in most departments, well, I think in probably all departments everywhere, and certainly in most institutions, the density of great science is decreased. The density of people who are doing science because as a calling has decreased, and it's been diluted maybe by a factor of 10 by people who are doing science as a career.
Yes. And that creates a lot of isolation.
Well, it's isolation for those who want to do it as a calling.
Exactly. Exactly.
Because when
Exactly.
it used to be when you went to a meeting, and
Yeah.
remember the funding rates were much higher because science was so much smaller.
Yeah.
Um,
Yeah.
50 years ago, when you went to a meeting, everybody was talking about the science.
Hmm.
You'd have debates about the science,
Yeah.
and you went out to drink and dine, and you
Yeah.
talk about the science. And there was lots of bad science. It's not
Yeah.
that, it was all great. It's that people would honestly debate and discuss
Hmm.
science, good and bad, because they loved it and they wanted to
Yes.
learn the truth.
Yeah.
But if 9 out of 10 people now are in it as a career,
Hmm.
and they're worried about whose grants getting funded and who's
Yeah.
getting recruited to some other university and
Yeah.
who's involved in the latest scandal,
Hmm.
um, the discussions are no longer about the science.
You can even question the usefulness of conferences today.
Yeah, absolutely. And in
Yeah.
fact, I've been to very few in the last few
Yeah.
years.
Hmm.
Um, so the culture has changed.
Yeah.
And the quality of the decision making has changed because, and I've run some numbers on this,
Hmm.
the volume of reviewing
Hmm.
that's required to sustain the scientific establishment, I'm talking about grant proposals and journal articles,
Yeah.
has gone up exponentially. Why? Well, let's look at the grant situation.
Hmm.
50 years ago, you probably had one grant. It had better than a 50% chance of being renewed. It was good for 5 years, and you didn't feel the need or even the possibility of having more than one grant.
Right.
So every 5 years, a scientist would put in a grant,
Yeah.
and even if there was a 50% chance of it being renewed, then you might have another 5 years of funding, or you might have to resubmit.
Yeah.
But one grant gets resubmitted once, and then it's likely to be funded.
Right.
Now, most labs have to have 3 or 4 grants
Hmm.
because you're never sure, in fact, you're reasonably sure that the ones you're on are going to not continued.
Yeah.
And and those grants, rarely last for 5 years. In most of the funding agencies, they're more like 3 or 4 or 2. And the success rates for even the renewals are like 10%. So everything's constantly being resubmitted.
Yeah.
So if you just add up
Yeah.
the number of grant proposals that have to be reviewed,
Yeah.
you think about what the burden is on the people who are going to review them, because it takes hours.
Right.
So I, by my computation, you could have in 50 years ago, you could have the top 5% of the field review all the grant proposals and make what would likely be pretty good decisions because they were the top 5% in the field. They
Hmm.
really know the field. They knew the players. There was a relatively small number of players, and they were the best scientists, with making the
Yeah.
evaluations. The present system, given the volume of reviewing, you have to have more than 50% of the working
Hmm.
scientists involved in the review process.
Yeah.
Well, I'm sorry, it's a bell-shaped curve. The top 5% were two standard deviations away from mean capabilities. They were damn smart.
Right.
Once you get down to the mean,
It's a
and
chance.
you have those folks reviewing papers that they barely understand, it's not so
Uh,
pretty.
it's the same thing with journal review as well. So I ran some numbers, and what I found was that a large proportion of grant money is actually used to dig in the next grant rather than actually doing the work.
Mhm.
So it's
Well,
like,
that's really a recognition
yeah.
of the strategy that I said before,
Hmm.
which is if you get a grant, it's because you'd already done the work,
Yeah.
in which case you use the money, if you're smart, to do something completely different that you think will get you the next grant.
No, that's one part of it. The second part of it is that you can submit more grants if you have more postdocs. So instead of
Uh,
actually doing real research, you write proposals and proposals, a
of
significant
course.
amount of time.
Oh yeah.
So it's kind of a lost battle for almost everyone. It's a
Oh
disservice
yeah.
to science, in that sense.
I was
Uh,
very fortunate to
yeah,
spend the first 15 years of my career
yeah,
as an intramural scientist at NIH,
yeah.
which meant that I was in a lab that had a budget
Hmm.
and basically there was no grant proposal writing.
Yeah,
And that meant I could take on projects that had high risk and long payoff times. All
right,
this work we did on
right.
single unit recording from chronic preparations and naturally behaving
Hmm.
animals, it took 5 years to do each one of those experiments.
Yeah.
And, we didn't become productive for the first 10 years.
Hmm.
And that not sustainable. You can't do that now as
Yeah,
a faculty on a grant. So, you know, again, there are
yeah,
positives and negatives to every system.
yeah.
The reason I left NIH after
Hmm.
15 years is I wanted to branch out into other fields, partly because I realized that what we'd been doing there was played out, partly because I realized that what we'd learned had some clinical applications that I wanted to pursue, which were not what the budget for our
Yeah.
basic research laboratory was
Hmm.
capable of doing. Now, if you're in a university, you get to write a grant proposal to work in a new field. If you're
Right.
at NIH and you want to work on something your lab is not funded to do, you're
Yeah.
screwed.
Right.
So
That's very
left.
true. Also, I think we need innovation in the scientific ecosystem because right now scholarship is not rewarded at any scale, I feel, because all the monetary rewards or career growth is tied up to grant funding. So, for instance, the celebration now is about the funding, even though funding is a means to the end and not the end in itself.
Yeah, I
So,
sit on a lot of appointment promotion and tenure committees, and
yeah,
nobody ever reads the papers.
yeah, exactly. So, so even though the scholarship is supposed to be the end of things. So that's about it as well. I also feel that we should be awarding or rewarding, as such, uh, conservation of resources in science. So, for instance, the way current system works is that if I spend more money, irrespective of what I did with it, I'm celebrated, even though somebody else can be very spendthrift and actually produce amazing science with very little money. So right now we do not have any, weighting factor for that capability, which is a very important ability for a scientist.
Yep, we make decisions as bean counters. And
Yeah.
well, why do we do that? Well, partly because our institutions need the beans, and partly because the science is so complicated, it's actually difficult, even within your own field,
Do you understand?
to read other people's papers and understand their significance.
Right. Okay, so a closing question for you because it's we are approaching about 2 hours and time is precious. For you is so somebody starting, right now, let's say, have all the opportunity to be free and to decide, from a fresh perspective about the human body and physiology, and interested in movement. So now, how should they approach the problem of motor control, and what kind of technologies should they focus on? Should it be technology, start with a narrow focus on certain kind of signal, for instance, EMG, or should it be a multimodal, all you can measure approach to the body? And what kind of questions? Uh, should we start with a specific clinical population because we know, or we have kind of a, presumptions about what might have gone wrong, so we know where to start from, or we have applications, or should it be about basic understanding and a healthy human? So where to start a very fruitful investigation of human movement?
So I think it's important to, in a system as complicated as the nervous system, to be able to be competent at and able to combine experimentation with modeling. I think each one without the other is a dead end, but together they're necessary. You can't understand experimental data without having models to see if you can interpret and understand that. And you can't test models without being able to figure out and actually test them in their experimental implications. So in systems neuroscience, somehow you have to acquire skill with both and combine them. In terms of which experimental methodology, as I said earlier, if you pick one that's been hot for a long time, you're probably at the end of its productivity. The hot thing when I was a student was single unit electrophysiology, microelectrodes. And we spent 50 years exploiting the limits of that technology in terms of the kinds of data it acquires and the kinds of interventions that you might be able to do clinically, recording and stimulating, at the single neuron level. And I think we pretty much played it out and have reached its limitations. The field that feels to me the same way as electrophysiology did 50 years ago today is optogenetics.
Yeah.
Just enormously powerful. And it's going to have an arc that's similar. Decades of improvements in its capabilities, in applications of it to basic science and probably prosthetics that are going to be enormously important, very challenging, always, difficult to combine the science with enough capability in the technology to be able to stay at the cutting edge of a new technology while doing good science. But I think if you want a hot field that's at the beginnings of its arc, that feels like the one to me in the field of neuroscience.
Okay. And you see more and more people going to optogenetics and more labs,
Oh yeah.
incorporating that, especially in the animal model systems.
And that's involved, of course, development of whole new systems and whole new preparations. Of course, it's mostly currently limited to mice.
Yeah.
And what people have been able to do with behavioral experiments in mice is extraordinary.
Yes. I recently visited one of the labs where they have Parkinson's model in mice,
Yeah.
and the data is just amazing data, which it yields and what you can think of. That's true.
And we will. So
Okay.
for a long time, people couldn't imagine doing
Yeah.
those experiments in mice, and
Yeah.
said, well, they're
Yeah.
if it works this way in a mouse, what do we know about how it works in a monkey or a human?
Right. Yeah.
Especially if you're going to be looking at these subcortical structures that I think we've overlooked for decades. You're probably better off looking at animals that are more dependent on
On
those
those.
structures and not so highly overwritten by the cortical control.
Okay. And what about a human lab? So somebody interested in the human movements.
So the
Or
work that
yeah.
I'm doing at University of Queensland is all human experimentation
Mhm.
and rather classical electrophysiology of EMG. We happen to have a niche in which those are adequate technologies to study the thing we're trying to study. Uh, I think eventually nailing down how those subcortical structures work is going to require invasive techniques in other animals. One of my collaborators and former students, Brian Corneal, is working with monkeys at University of Western Ontario to do exactly that. Um, so, you, we are able to quickly do significant things that give us useful hints in humans, and they're laying the foundations for what's going to be a much more extensive development that'll eventually require other preparations and other methodologies.
Right. It needs a fairly open mind, both with respect to the model system as well as, the measurement techniques, compared to locking yourself up, with one of the model systems or measurement ways.
Yeah, picking the right system, both
Yeah,
the technology and the animal preparation and the behavioral task is absolutely crucial. And just doing what it is you're already familiar with is rarely the best way to go. So you constantly have to push yourself to look broadly at all those possibilities and pick the one that you think will be most productive rather than the one that you're most familiar with.
that's correct. So thank you, Professor Loeb, for a wonderful, session. Uh, I hope, I think that's like the tip of the iceberg of the profound knowledge you have in this field. And I hope to have you again on Beyond Phrenology, with more detailed discussions on more specific topics.
Well, I enjoyed it, Tudor, and look forward to further discussions, both public in the podcast and private.
Of course. Thank you so much and have a great day ahead.
Bye bye.